Hi, everybody. My name is Ravid Densen, president of the Israel Cancer Research Fund. Welcome to Brilliant Minds, our monthly series featuring interviews with some of Israel's leading and most accomplished cancer researchers. It is my honor to co-host today's proceedings with Dr. Nancy Maizels, the highly regarded, hardworking chair of ICRF's Scientific Review Panel, which has the daunting task of reviewing and scoring all the 150 plus meritorious funding requests we receive year in and year out. In a little while, Nancy will interview Dr. Ravid Straussman of the Weizmann Institute, an ICRF City of Hope grantee, whose brilliant lab studies the mechanisms that modulate the body's response to a range of cancer therapies, all with an eye towards developing better treatment options for cancer patients. But first, as many of you know, June is Cancer Survivor Month, and in recognition and appreciation of that fact, last week, I recorded this conversation with my friend, fellow ICRF board member and writer activist extraordinaire, the eternally optimistic Linnea Olson, a 14-year lung cancer survivor and veteran of, Linnea, I can't remember, 3 or 4 clinical trials. Her blog, Life and Breath, Outliving Lung Cancer, is a brilliant, poignant read. Linnea, thanks for joining us. I would not be a friend if I did not ask first and foremost, how you feeling?
Okay. You know, I am in my 4th phase 1 clinical trial right now, and it's one that has come with a whole raft of side effects. And of course, we're in a pandemic, and I wouldn't be a friend if I didn't correct you. It's been 15 years that I've been surviving lung cancer since April
Time flies.
2005. Yep.
So, but I can't help but imagine that COVID-19 has been a little bit of a bummer and a special challenge. Can you talk to that a little bit?
Yeah, I think for me initially to be starting a clinical trial and also beginning a phase of social isolation all at once was a particular challenge because nobody wants to go through this alone. And of course, everything has changed at the hospital now, where you can't even bring a caretaker or the person who's brought you to the hospital with you. You go up alone. And as I live alone, I was driving myself anyway, but at least I had the opportunity to have friends come visit. And of course, there was also the very warranted worry that my clinical trial itself could be at risk because so many have been. I mean, this is just a different world.
Can you briefly describe the arc of your now 15-year cancer journey?
Sure. I was 45 when I was diagnosed in 2005. I had been sick for a couple of years and aggressively pursuing a variety of doctors trying to figure out what was wrong. At one point, I was asked if I knew what a hypochondriac was. At another point, I was diagnosed with adult onset asthma. And it wasn't until I got a recalcitrant pneumonia that I finally had a CAT scan and heard those words I never thought I'd hear, that I had lung cancer. And because I had never smoked, I think it delayed my diagnosis by several years. You know, it was something that just wasn't considered. And at that time, I mothered to 3. My youngest was 7. I knew I had to live. So I did everything I could to ensure that, including having most of my left lung removed and 4 rounds of adjuvant chemo. And I was very, very hopeful I'd be cured. Unfortunately, my first scan post-chemo, the cancer had already returned. So I had to very quickly switch my focus from living without cancer to how am I going to live with it.
Yeah, I actually want to come back to the clinical trials in a little bit, but 4 clinical trials, 15 years later, can you talk a little bit about the critical role cancer research has played in your survivorship journey?
Well, it's not only played a critical role. I mean, I simply would not be here because back in 2005, there was so very little you could do for lung cancer. And my original oncologist actually said to me, it's kind of like baseball, 3 strikes and you're out. And I had already struck out twice. Radiation was not an option for me. So we started this period of watch and wait, where I'd go in every 3 months, and every 3 months I'd have more of what he described as schmutz in my lungs. And by the summer of 2008, I was very symptomatic again, had a biopsy, was restaged to 4, and I said to him, "Is it time to get my affairs in order?" And he said yes. And when I asked him how long I had, he said 3 to 5 months. So I essentially did, as did my family, the hard work of dying, of saying goodbye. And there was not even a glimmer of hope that there might be something else. But then this amazing thing happened, and that is that my restaging biopsy had also been submitted for further genetic testing and had come back positive for an EML4 ALK translocation, which had just been identified as a driver in lung cancer. And by chance, there was a phase 1 clinical trial opening at the very hospital that I went to. One other person had enrolled. They had quickly died, in part because of the drug. So here I am with this sort of impossible choice, but I knew for certain I would die if I did nothing. And I figured the trial might extend my life for a few months. So I said yes that day in the office. And on October 1st of 2008, I became the 4th person in the world with non-small cell lung cancer to take an ALK inhibitor.
Wow. You know, it is, as remarked, National Cancer Survivors Month. What's your message to the cancer survivors out there, some of whom may in fact be on this call?
Well, okay, first of all, I should do my little bit about the word survivor. I don't really like survivor because it has this aspect of been there, done that, and I'm doing it. It also, there's just this sense that it's a little bit disrespectful toward those who would like to be here who are not. It's a noun, so I prefer to say I'm surviving, which is a verb, you know, very much in active tense. And I think my message would be, don't think probable, think possible, you know? And I never would have guessed that I would be here more than 15 years after my diagnosis. And that son of mine who was 7 back in 2005, he's going to graduate from college in the fall. I mean, what a privilege. And I think I view cancer treatment almost like you're playing a card game, you know, and you just have to play your cards carefully and try to get as much time out of each one as you can, because thanks to cancer research, there is the potential for there to always be some other effective therapy just right around the corner.
You know, my friend, I don't like the term survivor either, particularly as relates to you. You're a thriver, and you're an activist, and you've made it better for a lot of people. So thank you for that. I know one of the issues you're focused on is more user-friendly clinical trials. So what's the issue from your perspective, and how can they be improved?
I think that clinical trials could be more humane, frankly, you know, and although I am so very grateful to have been a participant, I also say that it is not only my privilege, it's been my burden. And the reason is when you join a clinical trial, you're signing up for more blood, more sweat, more tears, more money. And you are the one that those resources will be drawn from. And even though participation in a clinical trial is an opportunity, clinical trials have a lot of issues with accrual, getting people to join, unfortunately with disparity. And that's not good science if you don't have a population that reflects the general population. And it is my contention that it's not just you build it and they will come, you build it better and they will come. And there need to be more supportive services, more more understanding of the barriers to participation and ways to make it both more attractive and easier for people to participate, because a clinical trial is a difficult thing.
Right. Linnea, thank you for your thrivership. Thank you for your support of ICRF. Most of all, thank you for your brilliant friendship. I think you know, me and my family, how high regard you're held in and how much we love you. And thanks for your advocacy efforts. So we're going to do the reverse. Now we're going to go from the bedside back to the lab. And with that, Nancy, take it away.
So I'm Nancy Maizels, and I'm really grateful, as I'm sure all of you are, to Linnea for sharing her story. You can hear more about Linnea from a wonderful TED Talk she's given, and she's there online for all of us. Her story is really a call to action to all of us to work harder in the lab, to work harder in the clinic, and to work harder to do everything we can in support of cancer research. The ICRF, the Israel Cancer Research Fund, has been doing this since it was founded in 1972, specifically funding research in Israel, where there are lots of great ideas, wonderful scientists, but not as much money as necessary. And since then, the ICRF has funded about 2,500 grants with about $72 million in all of the major cancer research centers and institutes and university in Israel. And this year, we're proud to be funding 72 different investigators with nearly $5 million of research support. This is the third in the series of webinars. We started them this spring. People are very interested, obviously, in the intersection of human health and disease and what we can do about it. And we decided to focus the webinars on the very, very close intersection, the interface between cancer and infectious disease. And in that context, I'm really delighted to have here with us today Ravid Straussman, from the Weizmann Institute in Israel, to tell you a remarkable and fascinating story that will have huge impact on cancer research and treatment. Ravid was an undergraduate at the Hebrew University of Jerusalem, received his MD, PhD there, did postdoctoral work there and at the Broad Institute of Harvard and MIT. And he's now a faculty member at the Weizmann, where he's been since 2013. So welcome, Ravid. Thank you for joining us. And I'd like to begin by asking you about your research, which is obviously the most important thing to you. Tell us about the work in your lab.
Absolutely. So I prepared a few slides. Maybe I'll share my screen. Just 1 second. Can you see my screen?
Yes.
Okay, perfect. So my lab is really studying cancer, and specifically we're trying to understand the complexity of the tumor microenvironment. And by that I mean that if you look inside tumors, as you can see here, for example, on the left, tumors are not just cancer cells piling up, but it's actually like an organ. So you can see here, these are the tumor cells in a human tumor. But you see there's blood vessels, and there's many immune cells, and there are many other components, nerves and stromal cells in the tumor. And for many years, we've been interested to understand how these other components that are found in the tumor can affect the response of the tumor cells to chemotherapy. And in one of the experiments that we published a few years ago, we demonstrated that if you take cancer cells on a dish, on a plastic dish, and you treat them with chemotherapy, it's easy to kill the cancer cells. But if instead you take cancer cells and put next to them just normal cells from the body, and then you treat with chemotherapy, the cancer cells survive. So there's something in the crosstalk between the normal cells and the tumor cells that can protect the tumor cells from chemotherapy. And a few years ago, we found that not only normal cells are present in tumors, but also one can find bacteria inside human tumors. So this was discovered by Lior Geller in my lab. It was published 3 years ago in Science. We looked back then at pancreatic cancer. We demonstrated that the majority of the patients, when you look inside the tumors, if you stain for bacteria, as you can see here, you can find many bacteria in these tumors. That's another slide, again, demonstrating in brown bacteria in human pancreatic cancer. And when you look inside in better detail, if you look at with electron microscopy, you can see that the bacteria are present inside the cancer cells. So here what you can see, this is everything you can see here is one cancer cell. That's a nucleus, and you can see bacteria inside the cancer cell. But what was even more interesting for us was the fact that we found that these bacteria can protect also cancer cells from chemotherapy. And I'm showing you one example here. These are mice that have tumors in them, and we can manipulate the mice to have bacteria in their tumors or not. If the tumors are just like in patients with bacteria, as you can see, the tumor grow under chemotherapy. But if we treat these mice with antibiotics and get rid of the bacteria in the tumors, then you can control the tumor growth and you can rescue this, you can cure this mouse. And we found the mechanism. What we found was that normally when you treat these specific tumors with a drug, in this case gemcitabine, the drug goes into the cell, into the nucleus of the cell, and it causes DNA breaks that kill the cancer cells. But we found that if you have a bacteria that lies inside the cancer cell, the bacteria pumps in the drug and inactivates it by cutting it into two. So the presence of bacteria inside tumor cells can protect them.
So, go ahead.
And
so tell us more about the recent, there we go, the recent science.
Yeah. So we just 3 weeks ago, we published another paper in Science about bacteria in tumors. And what we did here, we took a more broad look at this. This was done by Dvor and Ilana from my lab, Gary and Noam. And this was supported by the ICRF. And it was done with a joint grant that I got with Dr. Dan Raz from City of Hope. And what we did here, we looked at different cancer types and asked, so do all cancer have bacteria in them? And what are the roles that these bacteria have in different cancers? And showing you just 2 glimpses of this study, I would say that one thing we found is that every cancer type have different bacteria. So here you can see different cancer types. So brain cancer, bone, ovarian, pancreatic, melanoma, so skin cancer, lung cancer, breast cancer. And you can see that different bacteria are prevalent in different cancer types. And as I showed you before, the bacteria are not just everywhere in the tumor. Here you can see an example. The bacteria in red are only present in the cancer cells inside the tumor. So the bacteria really like to stay inside the cancer cells, kind of protected by them. I can show you here in a higher magnification and in 3D, each one of here you see cancer cells. So this is nucleus, this is cytoplasm. And in green, you can see the bacteria that are right inside the cancer cells, protecting them from chemotherapy. And here again, using electron microscopy, you can find them. And these bacteria are alive. So we can isolate, we can take biopsies from patients and isolate bacteria and grow them. And the last thing I want to say here, and then I'm happy to answer other questions, is that Aviva from my lab, Aviva Rotter, she championed really the latest project that was funded by the ICRF. And what Aviva did is really cross-talking with what we just heard from Linnea about her disease. So Linnea described that she had what we called an EML4 ALK translocation that caused her cancer. So EML4, it's a gene, and ALK is a gene, and we know that sometimes there is a genomic aberration that causes these 2 genes to kind of fuse together and generate a new gene that is called EML4 ALK. And this gene is the driver of these rare types of lung cancer. And as Linnea described, there have been some ALK inhibitors that can really extend the life of these patients considerably. And Aviva asked, we know that many of these patients, unfortunately, under treatment, they become resistant to treatment eventually. And we asked, do the bacteria that are found in lung cancer have something to do with it? So I'm showing you here an unpublished result from one of the screens that Aviva did. So she took these unique cancers, the EML4 ALK lung cancer. As you can see here, if there's no drug in black, the cancer grows in our lab very nicely. And if you add bacteria, it has no effect at all. If you add crizotinib, which is the drug that Linnea was treated with, it really kills nicely the cancer cells. But if you add crizotinib and bacteria together, the cancer cells are not sensitive to the drug anymore. So demonstrating that there's unique bacteria that can confer resistance, they can protect, again, these cancer cells, these unique lung cancer cells, from this new biological treatment or anti-cancer therapy. And what we're doing right now in the lab, together with Dr. Raz from City of Hope and led by Aviva, is really trying to dissect the mechanism, trying to understand how is it done, because we know that unlike in pancreatic cancer, here the bacteria do not activate the drug. We know that the drug is still active, even in the presence of bacteria. And we suspect that the bacteria secrete something that protect the cancer cells. And really, in these days, we're trying to completely dissect the mechanism behind that.
So this would help people who are resistant to treatment with this drug, because it might explain why the drug is not helping?
Right, potentially. So there might be more than one resistant mechanism. So maybe there's additional resistant mechanism, and I mean, some of them have been already discovered, and that's why there's a second generation of ALK inhibitors that can help overcome some of the issue. But we suspect that, I mean, we know that there are many bacteria in these types of cancers because we can see them. And now, based on Aviva's finding, we know that some of these bacteria can really protect the cancer cells from chemotherapy. So if we can better understand the mechanism behind it, maybe you can block it. And then the treatments will be much more effective to these patients.
So this is really exciting, Ravid, because it means that not only does the genetics of the individual determine the tumor or the genetics that the tumor has developed, but also influences from the outside, the different exposures and experiences someone has had in their life that bring the bacteria to the person and then to the tumor. So do you have any idea how a bacterium infects a tumor cell? Does it begin in a normal cell and proceed to make it tumorigenic? What are your ideas about this?
Yeah, it's a great question, and I can only partially answer it. In the lab, we have now more and more evidence that bacteria are probably also present in some of the normal tissues. So, you know, when I was in med school, we were taught that there's tons of bacteria on our skin, and there's bacteria in our GI tract, right? In our mouth, in our colon. But we thought that internally we're sterile. So if you look at someone's liver, you don't expect to find their bacteria, or in someone's brain or breast. But I think that in the last few years, work from our lab as well as from others, start to see some bacteria in places that we don't really expect them. So one answer would be that yes, probably there's some bacteria in the normal lung or in the normal breast or even in the normal brain. And when these cells become a tumor, they carry the bacteria with them. But when we also compare tumors to their adjacent normal tissue, we almost always find more bacteria in the tumor rather than in the normal tissue around them. So there is enrichment for specific bacteria, and not only more bacteria, but sometimes we see bacteria that are tumor specific. We only find them in the tumors rather than in the normal tissue beside them. So these bacteria may come probably either from the outside environment. So our lungs, of course, are exposed to the outside. One can think about the breast because of the ducts, or the pancreas that have some pancreatic ducts that go into our duodenum, or from the bloodstream, because each one of us has what we call transient bacteremias. So when one brushes his teeth in the morning, there's some bacteria that can get into our bloodstream. And usually these bacteria are being taken care of very quickly by our immune system. But if one has a tumor, and sometimes patients have tumors in them for years before they are aware of that, these bacteria can leak out of the vessels and go to these safe havens because the immune system is somewhat repressed in the tumors, and maybe that's how they get there. So there's different options by which bacteria can reach tumors, and I suspect that all of them are actually happening in patients.
So this is really fascinating. We have a question from someone in the audience, and that is exactly which bacteria were found, and can you target them with antibiotics along with chemotherapy to kill the cancer cells?
Yeah, that's a great question. I won't be able to go into the details of which bacteria just because these are long lists and they have been just published. But I can say that in general, that the majority of the bacteria that we find in tumors are bacteria that are known to be present in normal people, but we also find not a very small minority, but a minority of bacteria that were never described in patients before. Some of these bacteria don't even have a name, so we will need to name them soon because they were not found before. So this was very surprising for us. I guess that these are soil bacteria or bacteria from other sources that usually cannot really survive the specific conditions that we have in our body, but the tumor is a very different entity. For example, it can be hypoxic, or it has different metabolites in it. So maybe these bacteria can survive there. As for antibiotic treatments, one needs to be very careful. It's been established over the last few years that some of the bacteria that we have in our body are actually very good for us. And this is also true when you look at cancer patients. So for example, we know that having a wide diversity of bacteria in our gut actually has a good prognosis. And some of the treatments that we give to patients are to some extent dependent on having these large consortia of bacteria in our gut because they probably regulate the immune system. So if you treat someone with a high dose of very broad spectrum antibiotic, you can actually harm this delicate balance. So I'm not sure that giving high dose of antibiotics to patients would be the right way to go. What we're trying to do in the lab is to really find out which are the good bugs and which are the bad bugs, and design therapies that are more specific to the bad guys, not harming the good bacteria.
So if you were to look in the lung of a normal person, this is another question from the audience, would you find bacteria if you looked hard, and would they be inside the cells?
Yeah, again, great question. I can only partially answer that because this is something we're doing right now. I would say like this, that first of all, it depends where in the lung. So the lung starts high, and then you can go, the more you can go to the periphery of the lung, the less bacteria you find. In our lungs, there are very small alveoli that are responsible for exchanging the CO2 and the oxygen, right? That's what the lung is doing. And there are specific cells in these alveoli that are called alveolar macrophages. And one of their main tasks is actually to protect us against these bacteria. So when you look at lung from normal people, you find alveolar macrophages that are full with bacteria, but mostly dead bacteria, most of them. So that's exactly what should happen. So bacteria get there from time to time, but are mostly taken care by these guards that guard the sterility of our lower lungs. But in tumors, in lung tumors, we definitely find live bacteria inside the tumors. So did they really escape between these macrophages and went in the tumor, or did they reach the tumors from the bloodstream? We're not sure yet. And how many live bacteria are present in normal lungs? I still cannot tell. We need some more time to explore that.
So we just got a question in asking, do probiotics help or hurt this situation?
So up to a few months ago, I would probably tell you that I do not know. But in the last few months, really nice studies from mostly from MD Anderson Clinical Center and other places probably demonstrate that probiotics are actually very bad for us in the context of cancer, and specifically patients that are about to be treated with immunotherapy. So one of the last conferences that I joined before the Corona era was in Orlando, and Dr. Jen Wargo, a very good friend of mine from MD Anderson, presented really exciting data demonstrating that when she looked at her patients that got immunotherapy, those that were on probiotics actually did not do very well. And she told everyone that she is now recommending all of her patients to stop probiotics. And the reason probably is because, as I said, the high diversity of bacteria that we have in our guts is very beneficial for us as a cancer patients, but also not as cancer patients. It's very important for everyone. When you take huge amounts of the very same bacteria daily, you kind of interfere with this large number of bacteria, and this is probably not good for us.
So here's another question, and it's a really interesting one. Can a cancer cell recruit a bacterium to help it survive?
Yeah. So, sometimes we talk about cancers and recruit. I'm not sure if it's intentionally or not.
Right.
But there was a very nice story from the lab of Professor Gili Bachrach from Hadassah Medical School. I think it was 2 or 3 years ago. And they demonstrated that there's a specific receptor called gal-gal-nak that is present on cancer cells, and that some bacteria are attracted to these receptor on the cancer cells. So he was one of the first to demonstrate that there's something on the cancer cells that indeed attract bacteria to them. I can also tell you that in our lab, we use a lot of mice models of cancer. And when we isolate bacteria from patients and we inject them into the blood of these mice, they immediately home into the tumors. We can actually stain these bacteria with fluorescent dyes so we can look where they are. You inject them into the bloodstream, and minutes later, you find them all only in the tumor. So yes, I think that, I'm not sure if it's intentionally or not, but we see definitely that these bacteria are attracted to cancer cells. They go there, they go inside the cancer cells, and they find safe haven there.
So that's especially interesting in light of the fact that people have worked tremendously hard to target therapies to cancer cells, and they have great therapies and great ideas for targeting, and usually they haven't worked. And a member of the audience has asked, could bacteria be used as gene therapy vectors to take therapies to cancer?
Yeah, that's another beautiful question. And we have a team actually in the lab working on that. So what we did is, as I said, we took biopsies from cancer patients from their tumors because we were interested to get these bacteria that home to tumors. We tested these on mice, demonstrating that these bacteria actually get to the tumors. And now you have like a warhead that you can put in the bloodstream and it will go directly to the tumor. But it's a very sophisticated warhead because this is a living creature So you can load it with things like genetic, as you suggested, like genetic content. And these bacteria, once it's reaching the tumor, it can start expressing things that, for example, call in the immune system or secrete drugs, or it's actually a playground, a huge playground. And there's endless ideas and not enough students to follow this at the moment. So we were trying to, sorry?
Ravid is making a very subtle call to potential students and postdocs.
Oh no, the lab is full. I don't even have a space now. The lab was so crowded that half a year ago, I actually put 5 students in my room at the lab. So we're 6 of us together right now because there was no room in the lab. It's very crowded. But
I hope they're all working hard.
Yeah, they are.
Yeah.
Yeah, but this is again, so we had endless ideas and we prioritized what I want to believe are the best ones, and we're working on that. And the idea really, we're actually now in preclinical phase in which we take these engineered bacteria and we try to cure cancer by injecting bacteria into these mice, seeing these bacteria go to the tumor, and then do whatever we tell them to do. Fortunately, we have antibiotics in hand, so if everything goes out of control, you can always just give an antibiotic shot and get rid of these bacteria if there's some real infection. But usually we don't see infection. We see the bacteria going straight to the tumor, and then they can do whatever we tell them to do.
So we have another question that's relevant to how many cancer therapies are administered, which is, does radiation work better if there are bacteria present?
Again, a great question that we haven't really tested. I had a physician coming to the lab working for us in like half a year ago, and he said, let's infect cancer cells with bacteria and radiate them and see what the effect, but we just didn't get to do this.
Yeah.
But I must say, from the other perspective, that my intuition is that there will be an effect, because, as you know, radiation is working by generating DNA damage, mostly in the cells. And we, but mostly others, have shown the last few years that bacteria can also affect the DNA and its stability. So if you have these 2 agents that are known to affect the stability of the DNA, I would guess that there will be some interplay there, but we haven't tested it yet.
Okay. So let me peek at the other questions.
Maybe I'll just say in the meantime,
Yeah.
while you're speaking, that it's a really special opportunity for me to speak with this audience. I must say that we're trying to push forward the science in the lab. And usually we apply for grants, and like the ICRF grants, and when we get it, we can actually move the science forward, but we don't see the faces behind these donors and without these unbelievable organizations that make all this huge effort for us to be able at the very end to do the pipetting in the labs and try to do something. So I really want to thank, I'm not sure, I don't see anyone in front of me except you, but I'm sure there are people behind that we should thank. Yeah. So I really want to thank them for their support, because this is what makes us able to do this work, and we're really trying to make it not just to end in scientific papers, but we really try that at the very end it will reach the patients and make some beneficial effects on their lives.
So we have one more question that's fascinating, and then I'd love it if you'd share the slide that I sent you of your last paper. So it's a really interesting question from someone. In metastatic tumors, are the bacteria in the metastases the same as in the original tumor?
Yeah, it's a great question. I would say like this, that we have preliminary results demonstrating that the answer is probably yes. And I would say two things. There was one paper 2 years ago from the Broad Institute looking at very small number of patients with colorectal cancer. And they compared the bacteria in the primary tumor in a liver metastasis. And they demonstrated it's really very similar, implying that maybe they travel with the cancer cells to the new site. And we have a paper under revision right now in melanoma, demonstrating something very similar. This is not the focus of the paper, but we demonstrate in this paper, again, a small number of patients in demonstrating that the bacteria are the same in the metastatic, in the primary tumor. But I also want to say that we are now really working on the bench on a very large panel of patients for which we have both the metastasis and the primary tumor. So in a few months, probably we'll be able to give a better answer. You want me to share my screen?
Yeah.
So let me share my screen. I'll put
Ravid has one more slide that I asked him to show.
Yeah.
So this is the title page of his paper that came out in Science less than a month ago.
Wait,
It's a totally amazing, we can see it, yeah? We can see your desktop, but it's okay.
Oh.
It's an amazing, groundbreaking paper. My husband showed it to me when it first came online before I even knew I'd be talking to Ravid. He's a scientist too, he was taken with it. And as you can see, there are lots and lots and lots of authors, including Ravid himself. I stopped counting when I got to 50, and Ravid told me that there are actually 61 different collaborators here who've been generous in sharing their samples, their hard work, and their ideas. It's an international group of collaborators, but as you'll guess if you look at the names, many of them are Israelis or in Israel. And the ICRF is really delighted to be able to support not just Ravid, but some of the other investigators who are authors on this paper. And we'd like everyone who's listened and who's interested and wants to learn more about the ICRF or the work that we do to log on to the ICRF website. And I think we're going to be segueing back to that shortly, and you'll be able to see all of this. So for now, I'd really like to thank you, Ravid. This has been great. I'd like to thank Linnea, and I'd like to thank Rob, and I'd especially like to thank the audience for your interest and for your attention. Without people helping, nobody would be able to do any research. So this is extremely important, as Ravid has told you. And now I'd like to end the webinar by saying this is the last of the series of webinars in the spring. I hope everyone enjoys what's going to be an unusual summer, a memorable summer for all of us. And the webinars will return in September. So enjoy the summer, be healthy, stay well, and take care. We'll see you in September.